Thank you so much for coming today to hear about mountain biodiversity and a little bit about conservation and climate change. So just take a deep breath, sit back. We're going to go on a little ride thinking about the mountains around you and what lives in them. So the first thing we'll talk about, we're going to have three topics today. One is local and global trends in mountain biodiversity, so at this kind of scale of how things are distributed on mountains. Then we'll talk about how local mammals are responding to climate change, in particular here in Colorado on the mountains. And then thirdly, we'll talk about maybe some unusual organisms. So predicting how climate change might respond, these particular carrion beetles might respond to climate change. All right, so give you a little bit of a different background. So I grew up in California near the Sierra Nevada mountains and was outside a lot and became interested in montane organisms very early on. And so this has been a continuing theme in my life. And then, as Dan said, I was in the Peace Corps in Honduras. And for those of you who haven't been to that country, it's actually really mountainous. And so there was a lot of extraordinary mountain gradients there that I learned about tropical mountains. And that's what led me to do my PhD research in Costa Rica in the Tiburon mountain range. And then in 2007, a year before I moved to this department and to CU, I started studying in the Colorado Rockies. First in the San Juan Mountains, here in the southwest corner, and then into the Front Range. Initially, in most of my career, I've worked on mammals, but over the course of the last decade or so, we've worked quite a bit on moving into insects, and in particular, these amazing beetles called carrion beetles. And then beyond that, as Dan also talked about, gathering data across the globe. We've been studying, the lab has been studying how mountain diversity is spread across mountains all over the planet. And here's just some of the data sets that we have. These dots are mountains in different vertebrate groups. We also have this for lots of different insect groups, some lichens. And so we've been looking at these trends in biodiversity on mountains, and then how they're impacted by anthropogenic change, so things like climate change and also by habitat change. All right, so I'm going to ask you to participate a little here. So all of you have been in the mountains, hiked up the mountains, maybe drove part of the way, took a bus part of the way, and then hiked up. So when you think about your favorite organism, it could be a moth, it could be an elk, and think about where, or choose mammals instead of elk, but think about where on that mountain when you're hiking up you see the most species. So it could be plants, it could be insects. And there's four potential, well, there's five, maybe even more, but I'll offer you 5 different ways to think about this. So here is elevation increasing here on our x-axis. Y-axis is the number of species, so more species higher. So when you're hiking up the mountain, do you see the most species at the bottom and fewer as you get higher? Do you see a broad number of species, high richness across the bottom of the mountain, and then it declines with a peak, but not so much a decline towards the bottom? Or do you see mid-elevations as being where you would see the most species? All right, think about it. All right, so what do you think? How many? It's the decreasing pattern. Sydney thinks it's decreasing. All right, who thinks mid-peaks? Ah, okay, nice. How about one of these two in the middle? A few, okay. So I think it's probably a third, maybe a little bit here. All right, so that's our first topic, is to see if who of us is correct. All right, so historically, when I started in this field, people thought that that decreasing trend was the only trend that organisms showed on mountains. And this was based on a really highly influential paper by John Tierborg, where he studied birds in the Peruvian Andes. And what he found, and here you can see these are summed up, but different types of birds. He overall found that as he hiked up the mountain, he found fewer and fewer species of birds. And this really stuck in people's heads as scientists. And for the next several decades, 30 years, 35 years, people thought that this was true for everything, even though there was actually quite a bit of mixed information out there or other kinds of patterns. And so when I started working in Costa Rica on those Tiburon mountain ranges, this is what we found. So the black lines with the black dots, elevation increasing, species richness on this axis, and you can see we found very strong mid-elevational peaks in richness. So the small mammals in Costa Rica were predominantly at the middle elevations. Here in the Southern Rockies, these are 4 of our transects, again in the black with black lines and black dots. You can see that for the vast majority of these, they're also mid-elevational peaks for mammals here in Colorado. And then we started studying all these other groups. So here's carrion beetles, these beautiful beetles I'll tell you a little bit more about later. They also have mid-elevational peaks in diversity in most cases. Ants, here's our ants, mostly mid-elevational peaks, right? Some of these gradients are right outside. Grasshoppers, again, mid-elevational peaks. And then we looked at mountain data from around the world. And just to orient you here, there's lots of information, but these are those patterns I was showing you. Mid-elevational peaks are these bright blue lines. So you can see right off the bat that those, for some groups, are the tallest histograms. So small mammals, salamanders, moths, ants tend to almost always show mid-elevational peaks in richness on mountains around the world, not just here in Colorado or Costa Rica. And then it turns out that these groups that show much more variable patterns, like frogs and birds, tend to show mid-elevational peaks on mountains that have dry bases. So meaning, like think about here, think about the Southwest. It's arid at the bottom, and the most productivity, the most plants, biomass is at mid-elevations. And on those mountains, birds and frogs tend to show their highest diversity at mid-elevations. So it isn't true that all organisms show one pattern. They, in fact, show variability in patterns, unlike what we thought from those historical famous studies. But mid-elevational peaks overall are actually the most common on mountains around the world, and in particular here in Colorado, Southern Rockies, and the western mountains. So you might be thinking, what causes these patterns? So a few years ago, there was a paper in Science that asked, or listed essentially, and discussed the biggest questions in science that had been unanswered. And this patterns of species diversity, why we see some places on the planet with lots of species and other places on the planet with very few, is one of those unanswered questions. Now we know a lot more about the distribution of the diversity, but we're still trying to understand why. One of the things we found in our research and other people have found in theirs is that productivity, so the primary productivity on the planet, is really important. So here's just 2 maps, global maps of diversity. This is for all of the mammals on the planet, and this is animal or vertebrate diversity. You can see these 2 look very, very similar. So warmer colors means more diversity. And then this is a map of global primary productivity. So in this case, these darker colors are showing you where there's more productivity on the globe, and the warmer colors up here show you there's less. And if you just compare these, you can see there's an extraordinary correlation between high productivity and high diversity of mammals, high diversity of vertebrates. It's also true for insects, a lot of different organisms, not all of them, but a lot of them. So clearly, climate and hence productivity are really critical for underlying biodiversity patterns on the globe. This is also true on mountains. Here's just one example. Here, again, are some of our elevational patterns in diversity for mammals. And then here is the productivity pattern for the mountains here. And again, you can see there's a really high correlation. Of all the things that we've tested, all these different theories, this is the only thing that holds up, is that primary productivity is very strongly linked to diversity. But the problem is that the mechanism, like why actually productivity is producing more species or is able to harbor more species, is still an open question. So all the things that we've tested pretty rigorously don't shed good light on this, essentially. But we do know, if you think back to those different patterns in diversity on mountains, that it can't be just one thing, because mountain biodiversity is slightly different. With the high elevation that has the highest diversity is slightly different for each taxonomic group: ants, moths, beetles, mammals. So clearly there's something important about evolutionary history, the taxonomic traits of organisms that interact with the climate. So one of the things, though, that maybe is more important in this context today, in kind of a general talk, is the conservation implications of this mid-elevational peaks in diversity. So think about now where we preserve our habitats here in Colorado, say in national parks or state parks. Here's just some really pretty pictures of where we have national parks around the US, right? Do you notice anything specific here? What do you think? I see a few mountains. So we are really good at preserving the very highest elevations, right? So most of our protected areas are at very high elevations, but usually higher than this peak in diversity that I showed you for many of these groups. We oftentimes do protect other places at low elevation, right? But oftentimes they're places that we can't farm. And that's another thing about our high elevation parks. We can't use these as much for our general human uses. But there's a lack of mid-elevation protection. So this is not my work, this is by another author, but this is just elevation now on a standardized standardized scale going upward. And then the proportion of land area in these different regions of the world. And you can see in the US, North America, we are really good at preserving high elevations. Same with Africa, Asia, South America. Europe does a more decent job here. Ireland's a little different. But overall, I just want to have that as be one thing to think about when you leave, is mid-elevations are where we have the vast majority of species, and we're not really focusing on protecting those elevations globally, or even locally necessarily. The only park here, the national park, that does a good job of this is actually Mesa Verde National Park. Mesa Verde is almost all either low or predominantly the mesa is mid-elevations. All right, so now we're going to move into the second part of the talk, which is how is some of that diversity responding to a warmer, drier climate? And in particular, I'll focus on our mammal work here. So humans are rapidly changing our climate. So, we're changing temperature, we're changing the weather patterns, which change precipitation patterns. And on mountains, we see this in the amount of snow. We also see this in shrinking montane glaciers. But all the organisms that live on the planet, as well as on mountains, are experiencing this change in climate. And this is caused by us, right? Another mammal. So we are increasing our carbon emissions, which has increased temperatures. So when I first started working in Colorado, this is where I wanted to try to understand not just their diversity pattern, but how they are in fact responding to warming temperatures. And the way that you do this as a scientist is that you need a historical data set about how things were before the world was warming, that then you compare to what we see today, and that is how you detect whether there's significant change. So how we built our historical data set was using museum specimens. So these specimens are incredibly important all around the world. We contacted about 45 different museums. We had them let us know in a database how many species and specimens they had in Colorado, and they sent us all of that data. It was like about 60,000 individual records, and built by historical mammal trappers, early explorers, and then lots and lots of mammalogists. And including a lot of our data in the CU Museum of Natural History. And so we broke these data into two groups. One that was historical, so before 1980s. So the 1980s is when, or in the 1980s is when there was a detectable change in temperature. So before that is our historical data, and then we built a contemporary data set where we went out and trapped all over these two mountain ranges. And we surveyed all of these small and medium-sized mammals. So you might be asking yourself, why these smaller mammals? What about the bears and the mountain lions and the moose, which are introduced? And the reason is because rodents are the most diverse clade of mammals on the planet. So about 40% of mammal diversity are rodents, smaller animals. And the other reason is because they have unique elevational ranges. So think about your favorite big mammal, like a bear or an elk or say a raccoon. They live across the whole elevational gradient for the most part, and they can move up and down, do altitudinal migration in the winter and summer, so that their elevational range isn't really critical for knowing how it's moving because they're everywhere. So this larger diversity of smaller mammals that have smaller ranges on the mountains that can change is what we're studying. All right, so you might think, what are the expectations of warming? So you've all been hiking in the mountains in Colorado. You know that as you hike up, it gets colder. And so in a warming world, if you're tracking a temperature range that you would, and you want to stay in the same temperature range, you would track that higher. So the predictions would be, if this is your mid-elevational range for some random mammal, and this is pre-climate change, and it's warming up, and you're acting as if you're tracking climate's important to you, you would move potentially to this elevation. So that would be a shift in elevational range where you have about the same span of elevations, but now you're just at a higher set of elevations. Another alternative would be where, you have a larger range, say like our yellow-bellied marmot, you occur from mid-elevations all the way to the top of the mountain, and maybe then you might contract to just higher elevations where your temperature range is post-climate change. So those would be the things that we would predict, right? You might also predict maybe that locally they might go extinct, but you could predict other things as well that aren't tracking climate. So things like staying in the same spot and adapting there. Or you might actually do better in this warmer, drier climate, and you would show us a response that isn't tracking climate at all. All right, so these are most of where we worked on these 4 elevational gradients. You saw those in some of our elevational diversity patterns. And these are the organisms that we had good historical data to compare to good contemporary data. We had 7 ground squirrels, here's some of them, 2 tree squirrels, 4 chipmunks, 7 species of vole, these little nocturnal, mostly nocturnal guys, mice, wood rats, 13 species, and then 4 species of shrew. There's a lot more small mammals that occur on these mountains that we've trapped, but we didn't have really good historical data to have really strong comparisons. So these are the ones that we did our analyses on. And here's just a snapshot of those mountains and the data. So the historical data are in these black dots, circles, and the contemporary sampling are the stars. So remember, these are specimen data. These are on our elevational gradients, but they're also in areas that we've trapped all throughout this region. All right, now I'm going to walk you through how to visualize these data. So here on the x-axis is going to be a species, all the different species names. This first one I'm showing you is Tamias minimus, one of the most common mammals in Colorado. And then this is their elevational range, from the bottom of the mountain to the top of the mountain, well, to where habitat stops. There's just rock above this. And then the black line, the thick black line, is the empirical historical data. So that's where they were historically. And then the colored line will be where we caught them contemporarily. And then these little thin lines are where we did modeling to assess undersampling. And I won't go into all the details there, but that's we built Bayesian models that looked at the sampling across elevational gradients, the detectability of the organism, and their overall population abundances. And that said, okay, we didn't sample well enough down here, we're going to assume that they were there. So for these comparisons, then we're comparing the overall smaller lines between each historical to contemporary comparison. And if there's a large difference, then that's where we say they've shifted in a particular direction. And in fact, here is one of the most common species of mammal, and it's not doing what we predicted. It's actually shifting to lower elevations, expanding their ranges. So overall, these are the species that we have data for in the Front Range, right around you. And this broken up into these blues and greens, those species are responding as we would predict in some way by moving upward. And then these are doing things that we wouldn't predict of tracking temperature change. So in fact, here are upward shifts. So some things that are moving upward, this is our golden mantle ground squirrel, our Uinta chipmunk, for instance, pygmy shrew. Here's some that are contracting upward, masked shrew, water shrew, golden mantle ground squirrel. And then these where just the highest elevations are expanding and the lower elevations have not changed. So for instance, the Abert squirrel. And then we have some that aren't responding at all. They're just, even though there's a 1 or 1.5 degree warming in their range, they're staying in the same spots. And then some of these species, like our Mexican wood rat, our meadow vole, are actually expanding their ranges. So doing things that we wouldn't predict. And that was also what we found in the San Juans. is that there's actually quite variable responses to climate change on mountains right around us. We have responders, and we have non-responders, and we have those that are actually doing better. So if you add up all of this range shift, and for all of the species studied on each mountain, and you say, where are the most of the losses? Those are in the reddish colors. And where are most of the gains in range? Those are the bluish colors. And you project that on the geography, you can see that the red colors are at low elevation, where most species are losing their range, and gaining range at high elevations. And you can see that in histogram view too. And you can see that extinctions are across the board, because remember, species have different elevational ranges, so where they're losing at the bottom can be at different elevations. And remember, when we're talking about moving up, shifting up, contracting up, it's not like our yellow-bellied marmot's putting on a backpack, packing up its nest, and hiking up, right? What's really happening here is that the lower elevation populations are declining and disappearing, and that the higher elevations are gaining in population numbers and expanding in population numbers. So we use this way of speaking about shifting, but it's actually that the populations are changing. So the average shift upward across all the individual species that we studied was 130 meters or so. And this might be what you would expect when there's been about a degree and a half of warming. But some of these species are responders and some aren't. And so that's what our lab tries to understand is this is the information that will help us understand who might be more at risk to climate change and who might be less at risk to climate change by knowing if they're responding or not responding. And so what we noticed right away, for those that are responding, 75% of those that responded with upward shifts like we predicted were montane mammals. So meaning that they already lived at mid elevations or at high elevations, and that 75% of those species moved to higher elevations. And they didn't just move a little bit, they moved a lot, so about 350 meters higher. So think about this as being 1,100 feet, which could be like a quarter or a third of the mountain gradient. So really large, drastic changes. And the traits, the mammal traits that are linked to this shift appear to be cold adaptation. And the reason we think that is, A, they're montane, B, the vast majority of species that shifted higher, or the amount that shifted, was also related to their geographic ranges. So if you live in the Colorado mountains, but also occur at really high latitudes, so think about, say, a wolverine is a good example of this, right? Lives in the high Arctic, all the way through Canada, Alaska, down into the mountains, right? That's true for all of these species as well. The other thing that's true about these species is that we studied them at the southern edge of their geographic range. So this is where it's warming the most and where they're kind of at the edge of their physiological abilities. And so that's why we think these traits are linked to somehow to their physiology. About those that are more cold adapted, or some kind of physiology or some sort of traits that they have is making them more susceptible to responding to warming. So the overall take-homes here is that, initially people probably thought everybody's going to respond to climate change and they're going to respond negatively. But that's not the case, and we know that from the paleontology record with albeit slower climate change, that there were very different responses in the Pleistocene. Some moved up, some moved down, some moved sideways, some moved into deserts, some didn't. That's also what we're seeing now in the Anthropocene climate change. The other point I want to make, and this is to reiterate the biodiversity one, is that mid-elevations are critically important to protect if you are interested in protecting things from responding to climate change. And that's because they need those mid-elevations to be able to shift their populations or grow populations at the other edge. So imagine if all of the elevations, say, at mid-elevations, become so fragmented that things cannot move through them, then they're going to be stuck at some elevation. All right, so the next part of the talk then is, what do you do? How do we predict how organisms are going to respond to climate change if we don't have that historical data? So we have museum collections for insects, but we don't have the same resolution. We actually have more individual specimens, but we don't have that montane resolution that we did with mammals. And so the other group that we've been working on in the lab are these guys, carrion beetles. We've been building museum data sets for them, but turns out we don't have a good enough set of data to say in these mountain ranges to compare their contemporary data to their historical data. So what we want to do with these guys is to understand their actual elevational ranges, how they're tied to their physiological niche, so temperature and desiccation, as well as their traits that are tied to those aspects, and then try to predict, given those traits, how they might respond, knowing there's going to be responders and non-responders. But we have to step back a little here and ask, why should we care, right? We never have to say this with mammals, because we are mammals, right? And we love our warm and fuzzies. But I'm going to convince you that you're going to love carrion beetles. They are very, very cool. So the first thing, though, to note is that, in general, my lab is interested in overall trends in biodiversity. And beetles are the most diverse organisms of animals, like terrestrial animals on the planet. So if we want to know something about biodiversity and its conservation and its response to climate change, we should probably know something about beetles. The second thing is that these beetles are key for why you don't see stuff dead on your hikes for the most part. It's a rarity, right? Or in your backyard. There's constantly things dying all around us, but we rarely see them unless we see them right when they strike a window or they're on a road. These guys, that's one of their main roles, is to clean things up and recycle it for other organisms. All right, and they're very interesting. And I'm going to explain why. Because this part of the study, the talk is a little bit about, wow, natural history is amazing. All right, so the family that they're in is Sylphidae. There's two subfamilies, the Sylphinae and the Nicrophorinae. So the Sylphinae are mostly black. They have a little bit of color, but not as much. They're kind of plain, and they're really small. And then the Nicrophorinae are these really beautiful Charlie Brown beetles. They're a lot bigger and colorful, and you can actually see them during the day occasionally, if you're so lucky to have a dead animal around you. All right, so those are the two main groups. These guys, their common name is burying beetle. So that'll give you an indication of some of their natural history. And you might ask, why does a mammologist work on carrion beetles? Well, they actually eat mammals. And the vast majority, they eat all carrion, and mostly vertebrate carrion, although some of them will eat decaying plant material. You can also find them in your outhouse. But in the mountains, the vast majority of biomass of vertebrates is mammals. So they're mostly focused on mammals, and this is how we found them in the first place. All right, so a little bit about their natural history. They like large carcasses. So this is a moose up by Nederland. And we were showing students these and collecting them, although I was the only one that would collect them. The sylphanae, the kind of plain ones, they reproduce on these big carcasses. So they'll eat any carcass, but they reproduce on a bigger one. So they'll mate, they'll lay their eggs next to the carcass, and they leave those eggs to develop into larvae that then populate that carcass and then do it all again. Where it gets weird is that the Nigrophorus, these orange and black beetles, they actually, for reproduction, use small carcasses. So things that are about 200 grams or less. So like a mouse. And here there's two of them. One finds it as fast as possible. It sends out pheromones for mates, attracts a mate. They decide they're perfect for each other. They get underneath the rodent on their back. And so their feet are on the rodent. And they try to move the rodent together. And if the two of them can move the rodent, they move it until some place that they've decided they can bury it. And so that could be, 5 meters, it could be 10 feet, could be 2 feet. And then they dig underneath the rodent, and they make a cavity, and they, kind of like this illustration, and then they cover up the cavity, and there's an actual space in there, like that. Then they clip all its hair. They have these special microbiome in their saliva, which is antimicrobial and antifungal, that they put all over this, and they just make a little meatball, literally. And then they mate and lay their eggs next to this. And it gets weirder. They stay there until the larvae come out, and the mother and father will actually feed the larva until the larvae will eat on their own. And this, of course, is super unusual for insects. Mammals, this, par for the course in some cases, but for insects, there's only a few organisms that will do this provisioning of offspring and pair mating. And so once the larvae are feeding on their own, then the parents will leave and try to find a new carcass and a new mate and do it again. A little bit more cool natural history is that you might be thinking, what would have happened if those two couldn't have moved that mouse? This is where it gets even weird. So they will find another pair. So another burying beetle will show up, attract a mate, and then the four of them will get underneath it and try to move it and bury it. And if they can, then they will all bury it and all reproduce together. So a true mutualism. But there are cheaters. So in fact, there's some cases where the bigger pair will just eat everybody else. But, but it has to be that it works sometimes, or else no one would share ever. And the idea is that there's enough meat in the meatball to provision both of their broods. All right, so here is where we've found, and we have a similar map for the San Juans, but these are the species we have here in Colorado, and these are their elevational ranges. So we have one that's a high elevation specialist, a few that occur mostly at high elevations, and then quite a few at low elevations. Like I said, we don't have really excellent data for these historical contemporary comparisons. So what we're trying to do here is understand, why do they have these different ranges? Is that tied to climate? And then is that Or is it tied to climate through these different kinds of traits that they have, and that's going to be who predicts, who responds, and who doesn't. So, like mammals, we think maybe something physiological is going on here. But in mammals, we couldn't test that very well. We can't take all 26 of those mammals into the lab, have colonies that we then test. It's just logistically impossible. But it's much more feasible with beetles. So what we've been working on first are their thermal tolerances. So are those at high elevations more tolerant of cold and less tolerant of heat, and vice versa? Low elevation species tolerant of heat and not tolerant of cold. Or are these other natural history aspects more important? So, for instance, this is what I'll show you all the data, but think about it simply as like high elevation species should have different traits thermally and for desiccation than you would think for low elevation. And we thought that originally because when we studied their diversity, we found that they weren't everywhere. They're most diverse and most abundant in thick vegetation at kind of medium warm temperatures. And these are mid-elevations, of course. So they're most in this niche of thick vegetation, which probably means that there's something important about humidity. So one of my PhD students, Dr. Chloe Garfinkel, who's now graduated, she led the work on thermal tolerances. And we put these guys in little warm baths and cold baths, warmed them up. And then when they can't, we had a little thing in there, and if they can't turn over, if it gets too cold for them to turn over again, we say that's the temperature, that's kind of their maximum or minimum. And this is what those data look like. Temperature here on the x-axis, hot, really hot, cold, right around freezing, right, and quite cold. But you can see that all of these, they were still able to turn over and do just fine at really, really high temperatures, above 99 degrees. And for the most part, they did just fine, and this is all during the summer because they're inactive in the winter. They just fine at freezing. The two highest elevation species, our alpine specialists, did have the best cold tolerance. And one of the other ones did too. That this one did not. It's kind of the same as the rest. And then if you look at the low elevation species, they're totally overlapping in their thermal tolerances, which was quite surprising to us. So it essentially says they all can tolerate much colder and much warmer temperatures than they experience in their range. And actually, much warmer than what they were going to experience in the next 50 or so years even. All right, so then maybe it's desiccation. So what we did here is we built these little drying chambers or wetting chambers, and we put them in there with many sleepless nights and watched how long or how well they did at different humidities. And when it was wet, they all did just fine, and we just let them go because it was a very, very long time they were going to be in there. Desiccation, they didn't do quite as well. So here's almost all of the same species, and what you can notice right away, most species are very desiccation intolerant. So if they get dried out, they can't be very active at all and will die pretty quickly. We had two species that were really good, although variable, in how well they would desiccate. Here are two high elevation species, one of each. Here's 3 low elevation species, 2 low, or mostly low, and 1 high. So again, desiccation tolerance, like temperature tolerance, was really not tied to their elevational ranges at all, which was pretty surprising to us. In fact, this species, Gatula, is the species when we worked in Mesa Verde National Park, that 99% of our captures were these, and it was essentially a desert, and they're terrible at desiccating. So all just very surprising. And from mammal world, where we were like, oh, it's all physiology, what we learned with carrion beetles is actually physiology is maybe not important as we think. Maybe they have much more broad physiologies, but what's important is how temperature and humidity is related to their life history and their life history traits. So we need to examine a lot more of those interactions between their environmental systems and their natural history. So some of the things that our lab is working on now is trying to understand how it might differ, these microclimates about burying versus not burying, big carcasses, little carcasses. We've been testing competition, we've been testing overwintering, and of course, the one key that maybe might be of interest to you is the microbiome. So think about these antifungals and antimicrobials. They might function well only in a certain temperatures or humidity regimes. And that might be where they're limiting their elevational ranges and hence their responses to climate change. All right, so we have talked a lot about conservation and climate change and things that are depressing and alarming. But remember that every one of us that cares about these things can do things ourselves. We have control over our local areas and our own actions. And so there's so many things you can do. You can just talk about these things. You can embrace alternative energies. You could take the bus a few times more than you do. So leave here thinking about the conservation and climate change implications, but also that we are empowered to do things about this to change the trajectory. All right, so lastly, I'd just like to thank a few people. So obviously all my funders and the CU and the Natural History Museum, the department, but also the people that nominated me for this award. So Becca Saffron in the back, Barbara Denning-Adams, I don't think she's here, and then Dan, of course. And then I want to thank my grad students and collaborators who did a lot of this work as well, many of them right here. And undergraduate researchers as well, who also have done a lot of this field work and lab work. And then lastly, my friends and family who supported me and helped me through good times and bad. So thank you so much, everybody, for coming, and I'm happy to take any questions you might have.